The common perception of mice is often negative. Mice are often associated with dirt and disease. However, scientific discovery reveals their capacity for joy. Mice also exhibit the ability to laugh. This challenges the traditional negative view of mice. In the 1990s, scientists noted unusual sounds from mice during play. These sounds were characterized by their high frequency. The sounds were inaudible to the human ear without special equipment. This discovery sparked initial scientific curiosity about the nature and purpose of these sounds. Specialized equipment is employed to capture the high-frequency sounds made by mice. These sounds are recorded in a controlled indoor environment. The recordings are then analyzed to identify distinct patterns. This analysis focuses on differentiating these patterns from typical distress calls. The goal is to isolate and understand the unique sound signatures of mice. This process aids in studying mouse behavior and communication. Acoustic frequency analysis is used to identify and categorize sounds. Specific acoustic properties are examined to determine the nature of a sound. In this case, the sounds are identified as laughter based on their unique acoustic characteristics. Contextual information further supports the identification of these sounds as laughter. Researchers conduct tickling experiments on mice to study social behavior. Tickling elicits high-frequency vocalizations in mice, indicative of positive emotions. The interaction also stimulates playful behaviors, such as jumping and running. These responses suggest tickling can be a valuable tool for understanding social reward mechanisms in animals. Sounds produced during tickling are analyzed and compared to other emotional expressions. The comparison focuses on differentiating tickling sounds from those associated with fear. The study also contrasts tickling sounds with vocalizations indicating pain. The analysis aims to determine if tickling sounds constitute a unique emotional expression. Results confirm that tickling sounds are distinct from expressions of fear or pain. This differentiation supports the idea that tickling elicits a unique emotional response. Brain scans can reveal neural activity associated with specific behaviors. Studies on mice show increased activity in reward centers during laughter. These reward centers are linked to pleasure and positive reinforcement. The observed brain activity mirrors that seen in human laughter responses. This suggests a conserved neurological basis for laughter across species. Mouse laughter is a form of vocal communication. This laughter serves as an emotional signal. It indicates the mouse is experiencing happiness. It also suggests the mouse is feeling enjoyment. Mouse laughter is analogous to human laughter. Both express positive emotional states. Young mice display laughter-like behavior during play. This laughter occurs specifically during social interactions with other mice. The behavior mirrors the joyful interactions observed in children. This suggests a potential evolutionary link in social behavior. Further research is needed to fully understand the nuances of this behavior. Mice exhibit positive emotional memory by associating places with laughter. This is demonstrated by their voluntary return to locations linked to laughter. The same behavior is observed with individuals associated with laughter. This suggests laughter creates a positive emotional imprint in the mouse's memory. Therefore, mice actively seek out environments and individuals connected to this positive stimulus. Laughter intensity differs among individual mice. These differences reflect variations in personality. Emotional disposition also contributes to the variation. Therefore, laughter intensity can be an indicator of individual traits in mice. This study focuses on observable behaviors in mice as indicators of psychological states. Mice subjected to stress or depression exhibit a noticeable reduction in laughter. In some cases, laughter may be completely absent in depressed mice. The presence or absence of laughter serves as a potential marker for assessing well-being. Reduced laughter suggests a compromised psychological state in the animal model. This behavioral change can be quantitatively measured and analyzed. Scientists utilize mouse laughter as a metric to evaluate antidepressant effectiveness. This method involves observing and quantifying changes in mouse laughter. Key parameters measured include the frequency of laughter events. The intensity or volume of the laughter is also carefully assessed. These measurements provide insights into the drug's impact on emotional and neurological responses. Research indicates that mice possess emotional capabilities. This challenges the traditional view of mice as simple experimental subjects. The recognition of mice's emotional lives raises ethical questions. It prompts a re-evaluation of how mice are treated in research settings. Ethical guidelines must adapt to acknowledge the emotional complexity of these animals. The neural basis of laughter in mice shares similarities with that of humans. These similarities suggest a common evolutionary origin for laughter. This indicates that the neural circuits involved in laughter may have been conserved across species. Studying laughter in mice can provide insights into the neural mechanisms mechanisms underlying human laughter. These findings highlight the potential for comparative neuroscience to elucidate fundamental aspects of behavior. Mice exhibit varying levels of laughter depending on their social environment. Mice living in healthy social groups demonstrate a higher frequency of laughter. Isolated mice, conversely, laugh less frequently than their socially integrated counterparts. This difference underscores the significance of social interaction for emotional well-being in mice. Healthy social interactions appear to positively influence the emotional state of mice, as reflected in their increased laughter. Isolation in mice leads to a reduction in playful behavior. This behavioral change indicates a negative impact on their well-being. Psychological health is adversely affected by solitary confinement. Social enrichment is crucial for maintaining healthy behavior patterns. Providing social interaction helps mitigate the negative effects of isolation. Laughter in mice is an indicator of their perceived safety. Mice only express laughter when they feel completely secure. This suggests a direct correlation between environmental safety and emotional expression in mice. A secure environment is crucial for mice to exhibit signs of joy, such as laughter. The presence of laughter can be used as a measure of well-being in mouse populations. Absence of laughter may indicate stress or a perceived threat within their surrounding. Mouse laughter is now recognized as a key indicator of well-being. This vocalization is used to assess their quality of life. The presence of laughter suggests positive emotional states. Its measurement promotes improved care standards in laboratory settings. Better care and treatment are facilitated through this welfare assessment method. Mice, despite their small brain size, exhibit a range of complex emotions. This emotional capacity challenges the anthropocentric view that complex emotions are exclusive to humans and larger-brained animals. Research suggests mice experience emotions such as fear, joy, and empathy. These emotions influence their social behaviors and responses to different stimuli. Studying emotional complexity in mice provides insights into the neural basis of emotions across species.